So for part 2, I'm going to start with this slide here again. That was the last slide also of part 1. And just to recap how hydrophobic signals work. You will appreciate them once we do the water-soluble ones. You will appreciate how easy the hydrophobic signaling molecules are. So start out here one more time. We have a lipid-soluble messenger. It is carried in the blood, typically with a binding protein, some lipoprotein that masks the lipid properties of the messenger. These messengers have no trouble crossing the phospholipid bilayer because lipids and other lipids, they like to mix. So they will cross the phospholipid bilayer of the plasma membrane as well as the nuclear envelope easily. They will find their nuclear receptors in the nucleus of the cell, bind to it, and then they become transcription factors that bind to DNA and alter gene expression in the cell. So here's the mRNAs and then the proteins that are resulting from these mRNAs. The messengers we're talking about here are all the steroid hormones. So that would be the sex hormones, testosterone, estrogen, progesterone. Then you have cortisol, the stress hormone. And then you have also aldosterone, a mineral corticoid. So all of the steroid hormones, plus, very importantly, thyroid hormone. Thyroid hormone is not a steroid, chemically speaking, but it is lipid soluble and it works just like the steroid hormones. Okay, so now we're ready to move on to the water-soluble messengers. And so here, kind of simplified, what you see here is these are signaling molecules that have to bind to a cell surface receptor, this thing right here. So there's some sort of a membrane receptor, and these water-soluble signaling molecules, they need to bind to their appropriate receptor, and then they will take that, they will cause a change to the receptor itself, and that will carry then sort of the signal into the interior of the cell and causes a cellular response. The response is typically faster as opposed to the steroid hormones, the lipid-soluble messengers. It is also usually involving some sort of a signal amplification mechanism. They're fast and effective. Trouble is that once the signaling molecule binds to the receptor, there are many different options of how this can go. And these are known as signal transduction pathways, and they get a little bit complicated. So this here is a simplified diagram showing how these water-soluble messengers must bind to a cell surface receptor. So then let's take a look at some of the options. And I'm sorry to tell you, these are just some of the more important options. Maybe a little bit of good news is that out of the 4 that are shown here, these major pathways, we're going to take only 3 for the purpose of this class. So what you see here, number 1 option would be that the signaling molecule, the hormone or messenger, binds to a receptor that is an ion channel. And in response to the binding of this signaling molecule, the ion channel then opens or closes, depending on the situation. So very direct kind of mechanism. The signaling molecule binds to this channel right here, and that alters the channel functioning and the flux of ions through this channel. Good example for this is acetylcholine binding to the acetylcholine receptor at the neuromuscular junction. That opens an ion channel, cation channel, and that carries then the signal into the interior of the muscle cell. Here, the next scenario is this one right here. These type of receptors are tyrosine kinase receptors. So they the receptor itself, it will bind to the signaling molecule, of course, and then the other part, the more intracellular portion of the receptor, is an enzyme. And it's typically a tyrosine kinase. That means it phosphorylates other molecules by their tyrosine amino acids. And these type of receptors are very typical for growth factors and some other signaling molecules that could bind here. So platelet-derived growth factor or any of the growth factors, they operate through these types of receptors. on the intracellular side, so this part of the receptor is an enzyme that then phosphorylates other proteins, thereby turning them on. This pathway is really important, but for the purpose of this class, I have to say that the next one is much more important than this one. So these an enzyme type of receptors, they're very important, but for the purpose of this class, the big number one favorite should be this one right here. This thing is called a G protein coupled receptor. And you should really pay attention to how they work, because in physiology, the vast majority of receptors that we're talking about are all G protein coupled receptors. Let's take a look at those. So these a G protein coupled receptor is a seven transmembrane domain type of receptor that's then linked to a G protein. So the G protein is actually this thing right here. The G protein has 3 parts to it, and they're all different. That's why it's called heterotrimeric G protein. The receptor has 7 transmembrane domains. So if you count those, how many times does the protein itself go through the membrane? And you see 1, 2, 3, 4, 5, 6, 7 transmembrane domains. So the receptor itself has this type of arrangement of 7 transmembrane domains, and then it is on its intracellular side, it is linked to a G protein, a heterotrimeric G protein. So what happens when the hormone or the signaling molecule binds to this type of receptor? So here the thing binds to this receptor, and that will cause a change, a conformational change that sort of kicks that G protein up, usually a portion of the G protein off the receptor. And then the G protein shoots off typically either the alpha or the beta gamma subunit, and it will activate whatever its next door neighbor is. And that could be an adenylate cyclase, it could be a phospholipase C, and there's a variety of different options how this can go. These type of receptors have enormous versatility. Pretty much all of the chemoreceptors that you have in the human body operate as G protein coupled receptors. Almost the entire sensory system operates on G protein coupled receptors. Your nasal epithelium, all of those chemoreceptors that are capturing odorant molecules, they all are G protein coupled receptors. We have thousands of them in the human body, and they are very important for physiology. So we're going to focus a lot of time on how this thing will work. The integrins are important too, but for the most part, they are linked to the cytoskeleton, and they are responsible also for cell linkages. So they're binding tissues together, and for the purpose of this class, we will not be discussing these very much. So our number one favorite is going to be this one right here. Our number 2 favorite is going to be this one right here. And our number 3 favorite for the purpose of this class, I'm not saying that they're not important, it's going to be this one. And this one we are not going to cover for the purpose of this class. In your book, you will find a diagram like this. I'm not a big fan of it. It shows just in general that there's some sort of an external signal, and then here you have a radio that has sort of the receptor for the signal for these radio waves, and then a transducer that then translates that signal into something that you can hear out of the radio. And we also have amplifiers, and then the response here would be that the sound waves are exiting the radio. I think we should just take it right to the situation that we actually find in cells, where your signaling molecule, of course, is the first messenger right here. So the first messenger is binding to this receptor right here. And keep in mind that the first messenger in this case here never enters the cell. It's different from the lipid-soluble messengers. Once the first messenger binds to its receptor, then something happens to the receptor, and that will carry the signal then on. Typically, we have second messenger systems available that operate through cyclic AMP or IP3, and you will see there's a variety of different options. A lot of times you have an amplifier built in to where an enzyme gets activated, and then you have many more output molecules that then trigger the response of the cell. So let's take a look at a specific scenario. We're going to take the example of where an ion channel in the end gets activated. So here is your signaling molecule, and it's binding to its receptor, cell surface receptor, and then you're starting a signal transduction pathway. Very likely this whole system will involve amplifier enzymes. So these are typically kinases, or it could be adenylate cyclase, that will then form a second messenger that then activates more protein kinases, releases intracellular calcium, and ultimately you're going to get your cellular response. Alternatively, you could also open an ion channel that then alters the flux of ions that will change the membrane potential, and that in the end gives you your cellular response. This slide here emphasizes the idea of the sort of the falling of dominoes. You're starting out with your hormone or signaling molecule, and that activates one enzyme, and that activates another and another, and eventually you're going to have a roll-down effect all the way down to the nucleus, and then you have some sort of a product. Or it could also stay inside of cells. You might release some calcium, and that causes the cellular response. So here, the idea of the signal amplification. Here's your signaling molecule binding to its receptor. And then here you have what's called an amplifier enzyme. A lot of times it's either adenylate cyclase or some sort of it's an enzyme that produces either a second messenger or goes off and turns other kinases on, something like that. There's different options here available. The point is, once you activate an enzyme, the enzyme doesn't make just one product. It makes a ton of products. And then if these products then in turn turn on more enzymes, and they make even more products, and you get a huge output. So this whole idea of signal amplification is shown here, where one signaling molecule is binding to one receptor, but the output is huge. So let's take a look at this table right here. These are the most important second messenger pathways that you should know about. Mainly the first one right here, cyclic AMP as a second messenger, is going to be your most important second messenger that you're going to deal with. It's a nucleotide based molecule. It comes from ATP, so it's adenosine monophosphate, and it's called cyclic adenosine monophosphate because a chemical modification is done to the molecule that makes a cycle out of it. Anyway, so it's made from ATP, and the enzyme that's manufacturing the cyclic AMP is called adenylate cyclase, this one right here. And that one usually gets activated via a G protein coupled receptor. So this pathway is the most important one for you guys. You need to know this. Cyclic GMP is very similar, but for the purpose of this class, we're not going to talk much about cyclic GMP as a second messenger. Occasionally you will hear about it. It's linked to nitric oxide, and there's some other things. We'll pick that up in a different class. How about the lipid-derived ones? If your receptor, your G protein coupled receptor, if it is activating phospholipase C, so this one will be the enzyme, the amplifier enzyme, phospholipase C. Phospholipase C is an enzyme that will split phospholipids that are part of the phospholipid bilayer. It will split it into its head group, the phospho group, and then the lipid the fatty acid residual. So phospholipase C will pick up one of those phospholipids and just chop its head off, the head group being then IP3, and the fatty acid tail, that portion becomes diacylglycerol. So the two second messengers that are formed here are IP3, which is important, and diacylglycerol, which is also important, but you're not going to hear much about this one again. So IP3 releases calcium, and that's why we need to know this one. And lastly, calcium can work as a second messenger right here, and we will talk fairly frequently about calcium. Calcium does all sorts of things in cells, and you will learn much more about it when we do the nervous system.